Hi, I'm Hollis Woodard in the Department of Entomology at UC Riverside, and welcome to our video.
And thanks for joining the presentation. My name's Will Grover. I'm an associate professor in bioengineering at UC Riverside.
My research lab at UCR is focused on trying to conserve one group of our native bees, the bumblebees. So in general, we're trying to solve the problem of how we conserve and stop the decline of our native bee populations. Here I'm showing you a figure from a recent paper that came out of the University of Vermont that looked at counties across the United States and asked the question, where in the US are we asking for pollination services from our bees, but not necessarily providing them with the habitat that we need to support their populations? And one thing that might pop out at you is that the Central Valley in California and parts of Southern California are among the places colored in red where we are asking the most, we're depending the most on native bee pollination services, but where we are providing the poorest habitat. And so this is a problem that we're dealing with globally and across the US, but it's a problem, especially here in California, that we need to try to deal with. And the problem of native bee population declines threatens the health of our natural ecosystems, but it's also a problem for human food production. So native bees, like this bumblebee pollinating a blueberry flower, are very important in the production of many of the most nutritious food crops that we produce. And so the decline of native bee populations threatens our ability to produce nutritious foods that we need in order to sustain our human population. So one of the parts of bee biology that I'm most interested in is the early developmental period. So here I'm showing you a cartoon figure of a honeybee who you can see on the left is in the egg stage. And then as she develops, she turns into a larva, a pupa, and then finally emerges as an adult. So this process, this early developmental period, is very important for bee health because bees are very sensitive to the effects of pesticides during these early stages. And then we know that there are important nutritional effects on this process too. Nutrition also can influence things like whether a young baby bee develops into a queen or a worker in the social bees. And so this entire period is a fascinating period to work on. And there are a lot of interesting and important questions in bee biology at this stage that we haven't been able to answer yet. And one of the reasons why we haven't been able to answer these questions is that in the social bees, it's very difficult to rear bee larvae outside of the nest. So what I'm showing you in the figure on the right, this is a honeybee, and she's inserting her head into a cell that contains a larva or a little grub, an early developing bee. And what she's doing is she's inspecting these larvae, and then if they're hungry, she'll actually feed them. And so this is the process and how it's carried out in a social bee colony. But if we want to try to control and replicate this process in the lab, there are a few important challenges. So these adult bees, they're feeding these larvae by regurgitation. They're essentially emitting small amounts of liquid to feed these larvae. And they do this continuously. They feed them around the clock. And so if we wanted to try to do this by hand, we would have to tend to baby bees and mete out tiny amounts of liquid by hand across a 24-hour period. And that's something that we just can't ask anyone in our labs to do. And so what we really need to do is be able to control this process of larval feeding so that we can control what we feed bees, when we feed them, and try to answer some of these important questions about important stressors in the early development of the life of bees. In addition to this challenge of being able to control the diet of larval developing bees, we also have a lot of other questions that we're trying to answer in the lab about some of the threats that are facing bees. Some of these challenges occur while foraging. So bees are out collecting pollen and nectar to feed the brood. And there are some important questions about foraging biology as well that we haven't been able to fully answer.
All right, and that's where we come in. So I'll give you a little bit of background about me and my lab and what we brought to this project. So I'm in the Department of Bioengineering. And so at UC Riverside and at a lot of institutions, bioengineering, applying engineering practices to solve biological problems, it's often focused on medical issues, medical problems. And that makes sense. There's certainly a lot of challenging medical issues in the world around us. But, there's a lot more to biology than just human medicine. And so in my classes and in my interactions with students, like my students that you can see here in this picture in my lab, I'm always looking out for opportunities for us to apply bioengineering principles to other biological problems beyond just human health. And those problems may touch on human health, but they're not directly associated with patients and their health. And so I'm always on the lookout for those opportunities. And so this was a tremendous opportunity when I met Dr. Woodard and got a chance to learn about some of her needs and her research in entomology. And as well, we bring to the table a particular technology for bioengineering, and that technology is microfluidics. So if you've ever heard of it before, I'll tell you a little background on it. My student Haran there in the middle in blue is holding one of our microfluidic chips. You can think of these kind of like computer chips, except instead of having little circuits that move electricity around from one point to another, they have tiny little channels in them called microfluidic channels that can move really small amounts of fluid in any direction that we want, according to any pattern that we want. And so that's one of those. And so my lab's developed a number of these for different medical applications. And so the fun in talking to Dr. Woodard and learning about her needs was trying to think, hey, could we apply some of our technology in microfluidics to solving some of their problems involving bee rearing, involving feeding these bee larvae as they grow and turn into adult bees. And so the solution that we came up with is illustrated here. It's an automated system for feeding bee larvae. It is a microfluidic chip that we kind of affectionately call the bee brain that can generate in a very programmable way, almost like a computer, but it's a liquid computer. It can generate any recipe of food ingredients that you want and deliver those to the larvae that you're growing in a little conventional well plate shown here on the bottom of this picture. And so then on the left, you can see the individual ingredients going into the chip. You can see at the top the microfluidic components that do the mixing and prepare the recipe of food that you're interested in feeding the larvae. And then on the bottom, we see 96 larvae ready to receive their food. So here's an early prototype of this device that we've been working on. And if we zoom in on the middle of this chip, you'll see it contains several of these little circular chambers. Now, each one of these chambers is a microfluidic chamber that can, or it's actually a valve that can contain about a microliter of any liquid. And now a microliter is a millionth of a liter, right? So this is an exquisitely small volume of fluid. And moreover, the exciting part about it is that, as you can see here, we can contain a liquid like that little blue volume that I've shown there, but we can also control it. We can move it around anywhere we want. So we can move it down like that, or all the way to the left, or all the way to the right. It's kind of like a video game after a while. And so we can control all of these tiny microliter volumes, mix them together and send them in any direction that we want with really exquisite precision. Now that's useful in a lot of medical applications, but it's also useful, we think, in feeding bee larvae. And so here's a couple of videos showing our prototype larva feeding chip in operation. In this first video, we're going to fill from the top to the right with one food ingredient shown in blue. You can see that going on there. So those four little circles now are filled with one ingredient that we'll use. In the second part, we'll fill the four circles on the left with a different ingredient shown there. And then in this part of the video, we're able to mix and match and combine those two ingredients in any ratio that we want in the middle of the chip. So you'll see motion going on here. That's us taking different amounts of each of these two bee food ingredients and mixing them together and preparing them to be sent to a bee larva in the next step. And that's what this looks like. Here, the row on the left, we're going to empty those four chambers that you can see blinking right there. We're going to empty their contents out to a larva in the lower left. And then in the next video, another mixture of food is being delivered to a larva in the lower right, for example, in this demo. So here again, we're manipulating these tiny microliter volumes of bee food. We're delivering them to larvae in any recipe that a researcher like Dr. Woodard and her students would like. And more importantly, this is fully automated. So we're able to perform this 24 hours a day, 7 days a week, which is something that we can't expect out of our undergrads or our graduate students and wouldn't ask of them either. So it provides us with the automation that we need to automatically, hopefully, once this project is finished, rear bees for research purposes in the lab. And then the final part of the project was coming up with a little electronic brain, a little controller to run the bee brain chip. And so we made a very inexpensive, just a few dollars to make this, Arduino-based controller that you see here. You can peck in the volume of food that you're interested in feeding each of the bees, like 3 microliters, the rate at which, or the frequency at which you want to feed your bees, like every 6 hours, and hit go, and it will automate the feeding and control that little microfluidic chip that you saw on the last slide. And so, you know, the fun part about working with Dr. Woodard and her students has been, I feel like every time we meet with them and talk with them, we find even other amazing challenges and topics that they research where bioengineering could be useful in engineering and preparing a solution, a research tool for them to use in conducting their research. And I'll mention just one other example. They study bumblebees and how they buzz, and how that buzzing when in proximity to a flower can release pollen, as you can see going on here in this video. And so they mentioned that they would like to have a tool that would let them apply a certain buzz of a certain frequency to a flower and study how it releases its pollen. So after having that conversation, my students and I went back to our lab and said, we can build this. We know enough bioengineering to build a little prototype system, which is shown here. And this took us about 30 minutes to slap together in the lab. And this can apply using a little smartphone app that you can see on the right, whatever frequency or amplitude or duration of a buzz to a flower that you'd like, using this little nail or screw that you see there attached to a speaker in the middle. So pretty primitive, obviously still some work to be done. And yet this little prototype, which you can see operating here in this video, buzzing a tomato plant flower, is actually quite capable of replicating the kind of buzz that a bumblebee would apply to this. And here we see trying different frequencies until we finally reach a certain frequency right there, where the pollen payload is released from the flower in the same way that a bee would be.
So as you can see, we have a million questions about bees that we can use Dr. Grover's help with, and I don't think we'll ever run out of those opportunities. And we have a lot of fun having our students work with one another so that we can learn things about bioengineering, and they can in turn learn some things about bee biology. So thank you all for listening, and we appreciate your time.
Thanks, Dr. Woodard. And from my perspective, it's been such a treat for myself and my students to work with you and your students on this interdisciplinary project. You know, we're able to take what we've learned in our research and in our classes in bioengineering and apply them to something completely different than what we usually focus on in bioengineering, apply them to insects and their importance in the environment. And so grateful for the opportunity for the collaboration. And to that end, I'm grateful to the Delfino Agricultural Technology Research Initiative, which was the funding that actually got the collaboration between myself and Dr. Woodard running in the first place. So I want to acknowledge that research initiative for their support in making this collaboration between the College of Engineering and the College of Natural and Agricultural Sciences possible in the first place. Thank you.